Good morning or early afternoon, I guess. My name is Madison Cornwell. This is my final Beckman presentation talk, so wish me well. I've been working in the lab of Dr. Kenneth Kozik in neuroscience here on campus, closely with my graduate student mentor, Israel Hernandez. We are in the molecular, cellular, and developmental biology department here at UCSB. And I wanted to introduce you all to Alzheimer's disease through a series of pictures that are self-portraits done by an artist. The first one was prior to being diagnosed with the disorder. And through time, you can see how the progression of the disease has affected his ability to do his work and his portrayal of himself. So my work is relieving one of the symptoms of Alzheimer's disease in an Alzheimer's disease mouse model. So first of all, just to illustrate the importance of Alzheimer's disease in our country at least, 1 in 8 older Americans actually has this form of dementia. And it's also the 6th leading cause of death in the United States. And over 15 million Americans are providing unpaid care for individuals that are affected by Alzheimer's disease. And estimated costs are about $200 billion, and that's in the private arena of family income that's going into caring for these individuals, and also governmental expenditures. So it's a disease that's really affecting our country economically and socially, so it deserves our attention and research. So the two hallmarks of Alzheimer's disease, first of all, inside of the neuron, if you zoom into this yellow image right here, you have these neurofibrillary tangles, which are rare accumulations of a normal protein. And then you also have amyloid beta plaques outside of the cell. And this is also another rare accumulation of a protein. And I'll go and dive into a little bit more detail later if this sounds a little, too abstract. Presently, the disease is completely incurable. Current drug treatments only try to alleviate the symptoms by providing ample amounts of neurotransmitters inside of the brain. Basically, it's just trying to provide these neurons with all the materials to send chemical messages so that they're not limited by their resources, even though the actual processes in the neuron are lacking. And then on the left, we have a regular brain cartoon, and on the right, you see how the dementias cause massive cell death as a result of these two hallmarks. So a closer look at tau pathology. This is the intracellular neurofibrillary tangles that I just mentioned. So this is the one symptom of this disease that I'm targeting. So we start out with a happy neuron. This is what a neuronal cell will look like. And if we zoom into the axon, which is the long projection that sends a chemical message to the next cell, we find these short or long dynamic polymers of basically it's called the skeleton of the cell, called microtubules. And if we zoom into these microtubules, we find a protein called tau. This is the protein that I study in my lab. And what happens in the disorder is that kinases, which is a family of proteins, they will add this phosphate groups, which is just a chemical group for those of you with experience in chemistry. And then we call this protein hyperphosphorylated tau. Basically, it has more than one phosphate group onto the tau. And this causes the hyperphosphorylated tau to detach from the microtubules, which essentially causes disintegration of the microtubules as well, because it regulates their stability. So you have detachment of the hyperphosphorylated tau, which will then clump together to form these neurofibrillary tangles that we find inside of that are found, completely filling out the cytoplasm of the neuron, and we end up getting a lot of cell death, and this is what causes your dementia. So that's just a background on how the progression of this disease occurs. So our rationale was that if we could somehow block these kinases from adding these phosphate groups onto tau, then potentially, we want to see what would happen, and the rationale is that possibly we could eliminate this process altogether and keep our healthy cells, or potentially maybe even reverse the process. So our goal is to come up with a chemical compound that could be used as a drug to do this in vivo, and the first step is to look at a mouse model. And then the ultimate goal would be to reduce the amount of dementia that you find in patients over time after you administer this drug. So in my research, I use a mouse model, and these mice have been given 3 mutations that cause them to develop Alzheimer's disease symptoms, including the neurofibrillary tangles, which are caused by tau, and the amyloid beta plaque that I talked about in the beginning. But first, Because Alzheimer's disease is an age-dependent disease, I have to wait until my mice are very old and they don't remember anymore. So that's the first step. Next, I inject my mice with our compounds that were developed by a brilliant chemist in our lab. And then after 2 weeks of treatment, I extract the brain, take half of it, and do coronal slices in order to create these beautiful, thin portions of the tissue that I can create beautiful histological stainings. So and the first part I'm going to talk about is the histological analysis. So these brain slices should contain neurofibrillar tangles, or maybe they don't in the case of our treated mice. And so we want to be able to see these neurofibrillar tangles under a microscope. So our first step is to identify the hyperphosphorylated tau neurofibrillar tangles with our primary antibody. And an antibody is just a protein that's used in your immune system to identify any sort of pathogen. So in this case, we developed antibodies that recognize this hyperphosphorylated tau. And then with a secondary antibody that's tagged with a fluorescent molecule, we can actually see where this hyperphosphorylated tau is found within our slices underneath of a microscope. And specifically, we're zooming into the CA1 region of the hippocampus. And the hippocampus is a region in your brain that's responsible for memory. And it's responsible for the same things in a mouse as it is in a human, although we're a little bit more complex. So basically, from this process, I can get images that look like this on the left-hand side. So our blue staining is just identifying nuclei. So every single cell that you find in this little tiny CA1 region of the hippocampus, you'll see as a circle in blue. And then our red staining, which I've put on a gradient of intensity. So if the fluorescent intensity is very, very high, then you'll see it as a white, or to get in the yellow range, going down into red. And this is actually identifying hyperphosphorylated tau. So as you can see, in these mice that are a disease model, you would expect to see the neurofibrillar tangles, and in fact you do. In our untreated samples, which are it's the top row, you see several very bright white or yellow cells that are full of this hyperphosphorylated tau. Whereas in our treated samples, either in a low dose or a high dose of the first compound we tested, I can't tell you the name of it because it's on lockdown, we see we've almost completely eradicated the tangles from these brains. And of course I'm going to pick the best pictures to show you, but I'll dive into that complication a little later. So we also tested it on a second compound, which is basically the same original chemical structure, and we just modified a couple side groups. And we saw the same result. So in order to make all of you believe my pictures, I have to take every single picture that I have and actually quantify the area of the CA1 region, which would be circled in red, and the number of blue nuclei that I found in these cells. And it's a very painstaking process. I have to manually count every single cell. So from these two quantified data points or data amounts, I can generate plots that look like this that represent the whole span of my experience. So every mouse is represented in these plots. So on the y-axis we have nuclei, so the number that I just counted, over the millimeter squared, which would be the area of the CA1. And then on the bottom we have vehicle, which would be the untreated sample, and then the low and the high dose of compound 1 and compound 2. So from this graph you can see that our nuclei counts are not changing as a result of treatment. So my drug isn't toxic enough to kill these neurons right away. And oppositely, we're not saving cells from dying, according to this number. We're finding the same amount of cells in our treated as our untreated. Second step is going through my red pictures to identify the number of hyperphosphorylated, or the number of cells that contain this hyperphosphorylated tau signal. And then I use that number that I just quantified of the number of blue nuclei to generate a plot that looks like this, that normalizes the number of PHF1 positive, which is the antibody that I use to identify the hyperphosphorylated tau, over 100 nuclei. So in this way, we're normalizing this amount from picture to picture. And as you can see, for compound 1 and compound 2, treatment in general, not really sure what went on here, treatment in general will reduce the amount of cells we see that are full of this hyperphosphorylated tau signal. So the next part of my experiments are to take the other half of the brain and dissect and homogenize the hippocampus from this region. So the hippocampus is the same region that I zoomed in on in histological studies. And from this homogenized sample, so basically just suspended in solution, I can use SDS cage electrophoresis to input a well for every mouse and use an electrical current to move the proteins through the gel and separate according to size. And from this we can generate a Western blot, which will show us which proteins we have in the sample, and we compare densities to see which mice have more or less of this hyperphosphorylated tau. So I'll be able to get results such as this one. So on the far left, up on top, this band shows PHF1, which is that antibody I use to represent or to target the hyperphosphorylated tau, and I can quantify a density for each sample. And then I normalize that density over beta-actin, which is another protein in neurons and many cells, all cells. And this density should not change very upon treatment. So from these numbers, again, I pool all of the data from all of the mice that I've sampled to create a graph that normalizes the PHF1 over beta-actin of these density bands. And again, it's following the same exact trend of decreasing the hypophosphorylated tau in our treated mice. So future steps are to ensure reproducibility, and for my experiments I use 3 to 4 mice for each treatment sample. And so first step is to enlarge this experiment exponentially in order to convince the FDA that it's worth investing millions of dollars to run all of their testings in human beings. And the ultimate goal would be to see if this drug had the potential to decrease dementia of patients over time. So as an acknowledgement, I'd like to thank Israel Hernandez. He's a graduate student mentor who's been mentoring me since my freshman year. Dr. Kenneth Kozik is the PI in my lab, and he's provided, insurmountable support. That's not even a word. Dr. Kozik has provided me so much support over the last 3 years. And then Dr. Jume Singh is the chemist that came up with these compounds and enabled me to have a paper in my undergraduate year. And then also CNSI, Eric Lubin and Opie have walked me through every step since being a little baby here. And then of course Tyler and Maria for the summer program. Thank you.